Well, let me explain how the initial drug discovery process evolved. Our focus was to develop a drug which would be orally bioavailable. We hoped we could develop an agent which would be orally bioavailable, and at the same time we wanted to have a drug which would have a reasonable duration of action. Obviously, we wanted something that was not going to be ultra short so that it would have a sustained effect. And it turns out that TXA709, by virtue of its chemical properties, is actually quite ideal. It's very easy to formulate for IV administration. So it has the necessary properties that allow it to be readily formulated for infusion. In addition, it has great oral bioavailability. In our animal model studies, about 95% of the dose becomes orally bioavailable. So it has these two wonderful features that sort of have been designed and built into the molecule that work extremely well. And we've proven in animal model studies of infection that TXA709 is efficacious by both routes, whether it be by the intravenous route or the oral route. And there's some very significant clinical advantages to this. In the acute situation where you need to administer and have a rapid onset, it's ideal to have an infusible antibiotic. And TXA709 is capable of being such an antibiotic. In addition, after the infection is under control and you want to have a sustained effect on an outpatient basis, the fact that it is orally efficacious is also ideal, because now it's in a formulation where, again, the patient can take it at home at the required intervals. So we were fortunate in our design that it actually came to fruition, that these two special properties were able to be achieved with TXA709.